Hello everyone. So today I like to introduce my research article. The title is Antidepressant Screening Demonstrated Non-Monotonic Responses to Amitriptyline, Amosapine, and Sertraline in Locomotor Activity Assay in Larva Zebrafish. So my name is Michael Edbert Suryanto. I'm a master's student in Department of Bioscience Technology, Chung-Der Christian University, and supervised by Professor Chung-Der. So, depression is a common mental health condition associated with a state of sadness, hopelessness, and grief that may affect the thinking process, behavior, and feelings. Depressed individuals are likely to have low level of neurotransmitter, and antidepressant can help to balance those chemicals by acting as a regulator that inhibit the reuptake of one or several neurotransmitter. So antidepressant drugs has been used to treat depression and anxiety disorder for over 6 decades since 1950, and it has been proved to be an effective solution to treat major depressive disorder, social withdrawal, low mood, anhedonia, and other psychiatric symptoms. Literature reported the abuse of antidepressant drugs. Several cases were identified among individuals repeatedly using antidepressant without prescription or excess of prescribed dose. Since antidepressant has psychoactive properties that create a pleasant or excited feeling as well as elevated mood, this could be one of the reasons why people misuse antidepressant in order to get a temporary euphoria. Along with the psychoactive effect of antidepressant, they produce side effects which also can be fatal. So this is the example of antidepressant life-threatening effects cause, can cause a number of effects that are potentially severe, such as vomiting, insomnia, headache, weight gain, dry mouth, tremors, dizziness, and decreased libido. Some studies also reported that antidepressant can lead to more severe side effects such as self-poisoning and mortality, and even can increase suicidal attempts in humans. So the antidepressants actually have been divided into 8 different groups based on their mechanism. So this is the list of 8 groups of antidepressant, as we can see in here in this table, and we use 18 different antidepressants that already widely used for the medical treatment in here, and along in this table we can see also the side effect of the from its antidepressants. So the present study aims to generate systematical data for a deeper understanding of the adverse effect of various type antidepressant based on animal behavior of zebrafish larvae. Why we use zebrafish larva? Because it's extremely fertile and can produce progeny in large number, and also easily maintained with low cost and rapid reproductive cycle, and also it has relative small size, therefore the locomotive activity can be easily observed and measured in high throughput number. So furthermore, the zebrafish is make it a suitable model to test various drugs with neuroactive or psychotropic activity. So at first we test the 1 milligram per liter antidepressant exposure, but further experiment was conducted to observe the dose effect of this antidepressant by using several different lower concentration, which were this concentration. The overview of experimental design and the illustrated workflow that used in this study. So first, on day 0, the zebrafish embryo were raised in 90 centimeter petri dish, and on the day after 4 day post-fertilization, which is the day 4, the antidepressant were administered through the medium water of embryo zebrafish by using the selected concentration. exposure of 1 milligram per liter antidepressant. On the next day, uh, the zebrafish larva was individually transferred to the 48 well plate, and then the larva locomotion tests were conducted. Zebrafox Viewpoint, an automation tool and software to record and analysis the zebrafish larva locomotion. So there are three different endpoints that we can get, which is the total distance traveled, burst movement count, and rotation count. The locomotion assay were recorded in 80 minutes, consisted of 4 light cycle and 4 dark cycle with 10-minute duration for each cycle at room temperature. Better resolution for the adverse effect of antidepressant. The panoramic approach analysis was used to combine data collected from zebrafish locomotion alteration by using principal component analysis (PCA) and heatmap hierarchy clustering analysis. Of trajectories of locomotor activity in zebrafish larva during light cycle and during dark cycle. Generally, the locomotor activity of normal zebrafish larva during light cycle is lower than the dark cycle, as you can see in this figure's trajectories. Cycle almost or very small movement of the zebrafish larva from its well. So this is the well plate that used in this study. Total is 48 well plate. So inside this well, in single or one individual zebrafish larva were put inside. And during the dark cycle, as you can see, the zebrafish quite moving more actively than the light cycle. So this is the footage of locomotor activity of individual zebrafish larva in the 48 well plate. The result, comparison of total distance traveled, burst count, and the rotation count of zebrafish larva in light cycle and in the dark cycle. Of the antidepressant significantly reduced the locomotor activity compared to the control group. This was indicated by the blue asterisk color. As you can see, most of the antidepressant reduce the locomotor activity. 19 group, including one control group and the other 18 group of antidepressant. As you can see, most of them give a blue asterisk color, which means the antidepressant reduce the locomotor activity almost in every locomotor activity endpoint, total distance, burst, and rotation count. But on the other hand, some treated groups can elevate locomotor activity and display and increments were observed in several antidepressant treated groups, such as sertraline, duloxetine, fluoxetine, imipramine, amosapine, amitriptyline, and also display in the other parameter endpoints. After exposure to the antidepressant, decrements in locomotion zebrafish larvae were observed in almost all treated groups. This result makes sense because the antidepressant working by targeting neurotransmitter. Locomotor activity is a result active effect of antidepressant. The change in behavioral reaction indicate that the bodies are impacted by these psychiatric drugs. Behavior was also further demonstrated by the phototaxis response. As a result, that this antidepressant impact with the serotonergic activity, and this proof that the antidepressant can induce an anxiolytic effect to the bodies of animal of the zebrafish larva. Behavioral endpoints, burst count and rotation count also important to evaluate the movement orientation of zebrafish larva. So burst count was considered as a rapid movement which help evaluate the locomotion activity, which the sudden increase is a restoration of swimming movement is indicated as the startle response. Meanwhile, the rotation movement indicate the swimming orientation varied depend on various factors and can produce escape kinematic response depending on the stimulus. So this stigmatizes behavior is also crucial for studying anxiogenic and anxiolytic drugs. The chronological result of zebrafish locomotion activity during 80-minute recording. So this is the distance traveled, burst movement, and the rotation movement. So this is the from the first minute until the 80 minutes, which consisted of 10-minute light cycle, 10-minute dark cycle, and continue until total of 4 light cycles and 4 dark cycle. As we can see, generally the locomotor activity of normal larva during light cycle is lower than the dark cycle, and during the dark cycle it will increase the locomotion activity, and when it transition to the light cycle it will going to reduce again. Well, the black line, which is the control group, show a normal or the correct pattern of the zebrafish larva during light and dark cycle transition, which is give a down, up, down and up, down and up, down and up again during the light and dark cycle transition. Similar in the burst movement and the rotation also, as we can see in the normal fish, normal zebrafish larva, it will give a give a correct pattern, and it will give a beautiful pattern which will going down during light cycle and going up during the dark cycle. Similar in the left right rotation also same. However, several antidepressant treated groups did not show any remarkable difference between in this total distance, burst, and rotation movement in both light and dark cycle, which is in other words diminish the constant pattern compared with the control or normal zebrafish larva. As we can see from the most obvious one, the Sertarin group, which is it generally create not an up and down pattern, but it's just a normally almost to straight pattern, straight line. When we can see the example from the amitriptyline, the pink line, it almost diminish the up and down pattern. And the dosepin group from the blue line, light blue line, generate a lower rotation movement, which give no difference between the light and dark cycles. Evaluate the behavioral endpoint similarities triggered by different antidepressants, PCA, and hierarchical clustering with information collected from the locomotor activity alteration tests were performed. Result, there are three major clusters were generated. Here, the green cluster, cluster A, cluster B, and cluster C. The first cluster group was plausible since all of the antidepressant in here cause decrement in all the zebrafish behavior endpoint in the life cycle. Second group, this group is belong to the antidepressant that cause minor changes in the locomotor activity of zebrafish larvae. Since this antidepressant display a similar heat map pattern with the control group, as we can see in here in the heat map. In here, during the zebrafish life cycle, it will give a lower value because in here uh, in the life cycle, normally the zebrafish larva will have a low locomotor activity, and it's indicated by the blue color, which have a lower value. But during the dark cycle, it will give a higher value, which indicated by the yellow to the red color. Cluster group is display a most significant different result. As we can see from this heat map, the AMI and AMO, the amitriptyline and the amosapine give quite the opposite result heat map from the control group. As we can see, in the light cycle, they have a higher value, which indicate the elevated of locomotion activity. But in the amitriptyline group, it give a lower value during the dark cycle. However, in the AMO, it keep getting a higher value, which means it keep elevate the locomotion activity in zebrafish. Therefore, in the PCA cluster also display that this group have the most distinct position from the control. PCA and heat map results for antidepressant including AMI, AMI, the amitriptyline, sertraline, amosapine, uh, cause constant different behavioral effect compared to the most other antidepressant and also compared to the control. Thus, a subsequent study was needed to further observe these three interesting antidepressants. Antidepressants were selected, which is the amitriptyline, amosapine, and sertraline, which is caused the distinct effects in zebrafish larva locomotor activity compared to the other antidepressant tested. Investigation was conducted to further evaluate and examine their effects in different doses with the lower concentration from 1 ppb, 10 ppb, and 100 ppb. Graph from the graphic result, the antidepressant can induce or elevated locomotion activity after exposure with the low concentration of antidepressant, which is the lowest one is 1 ppb. It's indicated by the red asterisk color. From amitriptyline, amosapine, and sertraline in almost every locomotor endpoints that used in this study. And along with the increased concentration, some of them can reduce the locomotion activity, and it can obviously observe from the in the amitriptyline from the burst movement. It's getting lower along with the increased concentration of antidepressant, which is the 100 ppb. And in the amosapine also reducing along with the increased concentration. Similar with the sertraline also can reducing the locomotion activity along with the increased concentration. So there's a possibility that this antidepressant affected the locomotor activity by using a dose-dependent manner and produce a non-monotonic dose responses. Dose responses and MMDR was indicated by a clear biphasic pattern with inverted U-shaped curve displayed in the amitriptyline and amosapine treated group. As we can see in here, the concentration in the 100 ppb is getting lower, but in the 1000 ppb, which is the 1 ppm concentration that already previously measured, it's getting increased. So it's created a biphasic pattern that going up and down based on the different concentration that use. Similar in the burst and rotation movement, also it can elevate it in the lower concentration, but can reducing at certain concentration, but then again can increasing the locomotion activity at the next higher concentration. Differentiated groups, several different hyperactivity behavior were observed in the lowest concentration group, which was supported by the high level of burst movement and also the rotation. Even though their total distance traveled was similar level with the control group in both cycles, but they also display a biphasic pattern that can seen from this graph result. There are several key points of this study. So there are three important points. First, the antidepressant exposure can alter the locomotion with the decrement in locomotion in zebrafish larvae after treated with the antidepressant. But several antidepressants can induce hyperactivity response in zebrafish, which is the amitriptyline, amosapine, and sertraline. And after we investigate furthermore with different lower concentration, interestingly, it was displayed that the NMDR, the non-monotonic dose responses, were occurred on locomotor activity in zebrafish larvae, in which is they can decrease and increase zebrafish larva locomotion based on different concentration, resulting in a biphasic curve. Is proof that the behavioral approach is useful as an early screening tool to assess antidepressants effect. And overall, the result indicate that the majority of antidepressants produce anxiolytic effect with the lower locomotion activity, which is the hypoactivity in zebrafish larvae. But however, there are several antidepressants that can cause anxiety-like behavior in zebrafish, with display abnormal behavior with hyperactivity and altered phototaxis behavior. And after further evaluation and observation, we found that certain antidepressants such as amitriptyline, amosapine, and sertraline exhibited a biphasic effect, indicating that they can increase and decrease zebrafish larva locomotion in different concentration. And so this is the end of my talk. Thank you for your attention and thank you for listening.